I want to take you through bone therapeutics, highlighting to you that we're in transition, and you may have seen some of the recent communications. We are, like many of us, struggling in the current environment and making tough decisions while retaining value on our clinical assets and being able to deliver that value first and foremost for the patients, but also for those that have invested and supported us. Nevertheless, I'll take you through, and a lot of what I'm going to say is a good segue to what Sinata and Ross is achieving. The usual disclaimer. And so we're a clinical stage company. We have multiple indications that we aim to pursue, but we're focusing on fractures, and I'll come back to that with our main lead asset, a strong IP for our clinical asset, and also supportive of our platform, and we're listed in Europe. The proposition is MSC-based to bone regeneration with ALLOP, but we would aim at a certain point to go beyond on the usage of MSCs, very much in line with what Ross has mentioned in terms of improving the startup, the material from an iPSC, but also improving the function of what we can do today in gene editing. There's a very strong team. Some of these members will be transitioning out, part of our focusing activity, but nevertheless, the value is retained and is also supported by a strong international board. And the pipeline, as you can see, and this is important because, as Ross mentioned, and I stress, it's in meetings like this that we further our collaboration and seek opportunities for the assets that we have and sometimes may not able to fully exploit with the resources that we alone have, but together may establish. We had a failed asset, non-cell therapy, in osteoarthritis, so good for you, Ross, which is nevertheless able for partnering because it was not lack of efficacy, it was lack of distinction versus placebo in a challenging environment of managing pain. Nevertheless, a strong clinical asset for bone regeneration with ALLOP, and I'll come back to it, in more than one indications, and a pipeline of using MSCs that it's on full hold for the moment, but obviously there's opportunity to consider beyond. Our focus rests in ALLOP with a significant market opportunity that you can see there. The unmet medical need is there, and the market is an important one. I just wanted to give you a quick brief of what we put on hold, because there may be others here that we could engage in a discussion. MSCs are great cells. They've been used in an amateurish way. They are being professionalized in terms of better sourcing, and they need to be improved on the efficacy. We've done that already in terms of differentiating them towards bone formation. I think, again, what we need to consider to do is how to gene edit them to specific function improvement leading to specific therapeutic targets. And that's really the vision that we have, starting with ALLOP, now focusing on ALLOP only, but going beyond. I'm not going to labor too much about the advantages of iPSC. Ross mentioned that. I think it's important to say that there is the gene and the hypoimmunogenicity approach, again, by engineering these cells, by targeting them better to a purpose. ARM, ISCT, what we're going to be doing going forward is engineering cells. In situ, ex vivo, it's going to be playing with the cells with our gene editing tools to get them better soldiers, better effective therapeutic purposes. And MSCs are one of them, but there's multiple others. We have an asset that has started on that path. I re-stress, it's on hold, so if there are interested parties in the room, come to talk to me afterwards. Now, ALLOP, again, the market has invested, the market has expectations about a clinical asset that we have in a controlled clinical trial, back to the discussion we had before, and that's where we're going to focus, that's what we are going to concentrate all our resources to get to the significant clinical milestone and then see what happens beyond that. It is still not an MSE-based and is still donor-based, but has been optimized for cryopreservation and multiple. We've scaled it up already, and so we're able to weather some of the challenges of MSE production, and we've been successful on multiple regulatory discussions with that, understanding what the future is ahead for us. The value proposition is to accelerate bone regeneration, which we have seen in multiple clinical models, and you can see there on the left-hand side, correct? On the left-hand side, where we have in a long bone fracture, we have virtually doubled the speed of bone regeneration. So we aim to do that in a clinical study by overall reducing the time to heal and consequently reduce also late-stage complications, so delayed union and non-union. We have not only the preclinical evidence, but uncontrolled clinical evidence of tolerability and the ability to regenerate bone, actually inducing or re-inducing bone formation on kind of lazy bone fractures. And so clinical evidence, again uncontrolled, that we're able to in vivo, in human, do it, but also on other indications like spinal fusion. So clearly we've seen the cells to be tolerated and form bone. What we need to show is that indeed we do that above and beyond what is the normal healing process of bone, and we do have an advantage and we do bring a value to patients. So we're conducting currently conducting a controlled study in long bone tibial fractures, complicated tibial fractures, to really document that acceleration and that benefit. We're actually considering, and we'll come in due course with information on that, to really accelerate the opportunity of reaching the clinical milestone because of cash constraints, but also because of market expectations. But in any case, what we have to do, further on what we already have, is to do a controlled documentation of the evidence of the value that we bring, and that's what the study is ongoing. Strangely enough, we suffered because of COVID, because people were behaving properly. They were not going out on motorbikes or having accidents, so we got less fractures and less availability of the health staff to deal with these patients. So we had a dip, and some on the audience know the challenges that we have been through that. We had a dip on recruiting, which is now clearly picking up, and we're seeing not only sites being active, healthcare practitioners available, but really events re-happening, which is good because normal life is restarting. We do all of this with partnerships that we manage, and you've heard probably in the press some of the collaborations that we have ongoing, particularly with our Chinese partners, which has been fruitful but also challenging. It's an interesting discussion how multicultural interactions are sometimes relevant. Nevertheless, we seek with them an opportunity to continue to develop ALLOP, and we have additional partnerships that are more or less on hold, but nevertheless there for us to pursue some of the objectives that I mentioned to you before. What do we aim to achieve in the next steps? Clearly, we pursue and aim to accelerate and potentially even anticipate the clinical endpoint for our TF2 study. While engaging with the regulators, this is only a European study, so we will have to extend it to the US, not the study itself, but the next step. So the FDA is on our path. And so clearly finishing the study, regulatory interactions, and coming out with a plan for the phase 3, assuming the positivity of the TF2 study. That milestone that we aim to reach towards the end of this year, beginning of next year, is going to be a touchstone, is going to be a critical element for bone therapeutics going forward. Now, from a finance point of view, we have managed a special loan within the context of the European Investment Bank which is an important achievement, despite being alone, is alone in conditions that you may be familiar with, which are important for the development of the company. We have been able to raise in December, and we've just finished a transaction, or communicated a binding transaction last week. And so this is in fact enabling us, coupled with focusing our strategy, focusing our activities, reducing our costs, to be able to go on to deliver on TF2. While having discussions for allo partnerships, we're also very active on other M&A discussions, and really that clinical study is our objective. I should point out that we have a dormant platform that may be interesting to discuss with some of you during the course of this meeting. So we continue to have the vision of our differentiated IMSEG platform, induced, like Ross, gene-modified it, the next generation that we believe. We aim to accelerate the clinical development and deliver clinical relevant milestones with allo within the time frame that I mentioned to you. That is crucial. That is our focus. That's the only thing we'll be concentrating now. And we'll do that also looking at further collaborations that will help us achieve that. Here is the people you may contact. As I said, some of us here will be cycling out into new adventures, but nevertheless remain in control as we stabilize the company and put the company onto the path of that TF2 results. A final word. I've been in cell and gene therapy for more than 10 years. This is one of the most exciting fields to be in, engineering cells for the future of health of our patients. So thank you all for being part of that. And you know, I also have responsibilities at ARM and ISCT. Really, it's being part of the field, part of the company, part of a family that is delivering huge value to patients. Thank you for that.